Finally, we have reached proteins, the last biological molecule. The function of proteins are to act as enzymes, which help speed up or perform certain reactions inside of the body. They function in defense by forming antibodies. These travel through the blood, fighting off foreign invaders. They also function in transportation, as hemoglobin is a protein that carries oxygen throughout the entire body through the blood. They help us with support and structure. They're actually forming tiny little skeletons inside of your cells, and they help with movement, since protein is what your muscles are composed of. A single monomer of protein is called an amino acid. When we link multiple amino acids together, here's one amino acid linked to a second amino acid. They are connected by a peptide bond. When multiple amino acids are joined by peptide bonds, they form a string of proteins called a protein or a polypeptide. There are a total of 20 different amino acids. Each one has a slightly different chemical composition. Where the oxygen is located, where the hydrogen is located on that individual amino acid is going to change which one it is. How these amino acids are ordered in the protein is going to be the instructions on how to create a protein that's a hemoglobin, or how to create the antibody protein, or maybe how to create muscle. They're all coded for based on the order of the amino acids in this protein. The primary structure for protein is going to be this shape, like a necklace. It's a linear sequence of amino acids joined by peptide bonds. This is the first and called the primary structure of protein. We see it right here. A linear sequence of amino acids joined by peptide bonds. The secondary level is when structural patterns begin to form by hydrogen bonds. We'll see a picture of that next. Tertiary is a final three-dimensional shape of a protein that is achieved by all of those structural patterns and bonds. And quaternary is a molecule composed of two or more separate proteins that have joined together to form an entire structure. Here is the primary structure, that simple linear sequence. Here is the secondary structure. What you're going to start to see as that protein is produced, the linear sequence begins to find spots where it's natural for it to begin to bond together with the hydrogen bonds. Something was partially positive and something was partially negative, and it just naturally comes together. When it does, it forms either the alpha helix or the beta sheet. Either of these structures might only be on one section of the protein. If we have, for example, a protein that's built up of 200 amino acids, once we start to fold, one section, 1 through 50, might be a helix, and then 50 through 100 might be a beta sheet. Maybe 100 to 150 is another helix. So we can go back and forth between these, and it really alters the shape of each individual protein. The tertiary structure is whenever that protein has taken on a three-dimensional shape, its final three-dimensional shape, and it'll contain repeating secondary structures of that helix or sheet. A fibrous protein may be composed entirely of those helixes. It's very strong and you can actually pull on it. Globular proteins may just look like a big messy glob. You see both the helix and the beta sheet in these. Proteins are not just shaped willy-nilly. There is an intentional functional purpose for the shape of a protein, always. Sometimes a protein is forming a lock and key fit where it'll actually bind to a molecule and it has to be a certain size or to have an opening that's just wide enough for only that molecule or enzyme to fit. It has to be a lock and key fit. So the shape of the protein is essential to its function. If you mess up the amino acid coding by even a single base, It can change what the resulting shape of the protein is, and it may no longer be able to do the function that it was intended for. Some of our best supercomputers are still working on the mystery of how some of these proteins actually fold. So it's something that over the last couple of years that computer AIs have gotten better at understanding and interpreting what this code will be because it's so complex what shape it takes on. Lastly, we have the quaternary structure. This is when a molecule is composed of two or more separate proteins. Here in this example, this protein has formed from 4 globular proteins. 1, 2, 3, 4. This is actually probably a hemoglobin. It takes all 4 individual proteins coming together to form one hemoglobin. Here we see fibrous proteins wrapping together. This just increases the strength of this structure. One fibrous protein might not be that strong, but wrapping them together is a great way to increase the strength of that protein. How about we see this in action with the hemoglobin protein? This is a protein that carries oxygen in the blood. It contains approximately 600 amino acids. The primary structure is just that simple chain of amino acids. Amino acid 1, 2, 3, 4, 5, 6, 7, 8, all the way up to 600. Now, the order in that amino acids are specific to the hemoglobin protein. You will only see that specific order for this protein. When the linear sequence is complete, hydrogen bonds will form between the different sections, creating secondary structures like the pleated sheet or the helix. Those will come together and form the tertiary structure, the final three-dimensional form of that protein. But just a tertiary structure alone is not enough. We're going to combine 4 hemoglobin proteins into one structure. This quaternary structure is the entire hemoglobin. It's amazing that we have reached from just that linear chain of 600 amino acids, we got all the way to this structure right here, which is very complex compared to that linear chain. And with those 600 amino acids, you would think it'd be pretty flexible. Maybe if we got one coded incorrectly, it'd be fine. No, it's incredibly sensitive for that shape that it took on. There is a disease called sickle cell anemia that you may have heard of. Sickle cell anemia is caused by a change in 2 amino acids out of 600. Those 2 key amino acids are part of how it develops into a secondary structure. When they are missing, the shape of the hemoglobin is not correct. So whenever it comes together and forms its hemoglobin protein, it cannot properly carry blood because the shape of the protein has changed. It causes the blood cells to not be shaped properly. They become crescent-shaped or sickle-shaped instead of round. Because of this shape, they deteriorate much faster. They may only last 10 to 20 days instead of 90 to 120. It increases the amount of clumping that can happen in the blood as these red blood cells kind of stick together. And you have to inherit this change in the amino acids from both parents for it to be full-blown sickle cell anemia. But when it does, it's very severe. And it's only from 2 out of 600 amino acids being different in that chain. So it kind of gives you an idea of how sensitive your proteins in your body is to any changes whatsoever in those amino acids. When building those proteins, it took us a while to get from that linear to the secondary to the tertiary, but it's very simple to break those proteins back down to their linear form. This process is called protein denaturation. It is the loss of biological activity of a protein due to high temperatures or changes in pH. It can take that secondary and tertiary structures, and that high heat going to break all of those hydrogen bonds and take it right back down to that linear chain of amino acids. Enzymes are proteins that speed up chemical reactions by lowering the activation energy needed for a chemical reaction. Think of them like little machines that are being built out of proteins. We use these all over our body. Chemical reactions typically require high heat to run. If you've ever added sugar to cold tea that was unsweet, how did that work for you? Did you stir and stir and stir? And as soon as you stopped stirring, all of that sugar just settled down to the bottom of your cold glass? That's an example about how that low temperature made it almost impossible for the chemical reaction to separate that glucose. On the other hand, when you add sugar to hot tea, it almost instantly dissolves. The temperature sped up that chemical reaction. The thing is, though, inside of your cells, we don't want to get too hot. We have this nice temperature that your body maintains for homeostasis. A workaround that allows us to perform those enzyme reactions, but still not get too hot in the body are enzymes. Here's an example. This enzyme is glycogen synthase. Typically, when it comes to enzymes, they're going to end with the word ase, so polymerase, kinase, and most of the time they are kind of named for what they do. This one, you can think glycogen. Remember, that's the storage form for glucose, synthase, so it produces that. Its substrate is going to be a glucose monomer. The glucose monomer has a specific shape, and the enzyme is going to be built and function specifically for that substrate. Our enzymes have a lock and key fit to their intended substrate. That way, they're not just creating random chemical reactions all over the place. The glucose comes and binds and fits perfectly into that lock and key fit of the enzyme. And when it does so, it binds those two glucose molecules together, forming another section of that glycogen. Then it releases them and goes off to do it again. Grabs two more monomers of glucose, binds them together, lets it go, grabs two more monomers of glucose, binds them together, lets it go. So that's how we build the glycogen while also maintaining that proper temperature for the body. You have enzymes for many different chemical reactions all throughout your body, and they're all specialized for their substrate. This enzyme is not going to combine anything but glucose molecules. All right, that was the completion of Chapter 2. Chapter 2 was a pretty good review for all of those basic components. Now, you may have noticed that on our lecture, we don't really mention Chapter 3. Chapter 3 is an interesting chapter, but at the moment it's not too related to our concentration of AP, so we're going to just go over that briefly for a couple of slides coming up, and that'll be it for Chapter 3.